Good morning, all of you. How are you? Are you all fine? I hope so. Last class, we had done with our year. Then we know that what all is there, and it's very important, all those RER and SER, we have done with it. Then we know why it's called rough endoplasmic reticulum, because of what? Somebody said it. Because of what? Why is it called rough endoplasmic reticulum? Yeah, because of the ribosomes on it. Then I mentioned of the function of smooth endoplasmic reticulum, gave the structure, gave the functions I mentioned. And I've given notes on that portion, and somebody said, when I give diagrams, it is understood, you have to draw it. When you draw, you'll understand more better how an endoplasmic reticulum looks like, or Golgi apparatus, how it looks like. So, some of you just came to me asking, how does it look, or what it is. Should we draw it, or should we not draw it? You draw it. I am not expecting you to be Raji Ravi Verma or something. Okay, I don't want big high-funda drawings. Neat, labeled diagram. That's all what I'm looking for. It should be neatly done and labeled as well. Okay, and then we discussed about the Golgi apparatus. And we said that the main important point there is the Golgi, they help in the formation of our lysosomes. Then we went through lysosomes also. Okay, and the main attraction over there was lysosomes are, what is it? The main attraction there, they are known as suicidal bags of, right? They are known as suicidal bags. That also we mentioned. And if anything happens, or if a cell ruptures, or anything happens, any destruction happens in the cell, what will happen? The lysosomes obviously will burst out. Okay, so these are the things which I have, I've just recalled so that you know that whatever you have brushed up. If you have just read the notes, and if you have gone through this, you would have understood that what I have taught. Then now we will start with the next, and I know that where we have stopped, my dear mitochondria, my favorite cell organelle. I cannot stay without telling. I don't know why, I just love this organelle. Okay, and maybe now your favorites after hearing me say. So here we'll start today with mitochondria. Mitochondria, the main, every time we say, or last year you have been hearing every now and then, they are known as the powerhouse of the cell. Okay, they are known as the powerhouse of the cell. They are double-layered membranes, like you can see, they have inner and outer membrane. Okay, they have inner and outer mitochondrial membranes, in fact. So these certain points which I'm saying will come up in your notes, you don't worry about it. So here when I want to stress, make it easier for you to understand, that is why I've given like this. You can see the mitochondria as such, you can see it's a double-layered membrane, and it's an inner and outer membranal, okay, mitochondrial membrane. So they have inner and the outer. Now, in the outer covering, it is porous. When you say porous, it means exchange of materials happen in and out within the cell. And here you can see finger-like projections, or you can see that projections there, finger-like projections. From the inner folds, it's folded, and that's called cristae. Okay, it is called cristae. The inner or finger-like projections, you can see it is called cristae. Mitochondria, mostly you'll be drawing it like this itself. A little more better, I'll make it easier for you. So it has an outer membrane and an inner membrane. Okay, and they are known as the powerhouse of the cell. The double-layered membrane, inner and outer mitochondrial membrane. Okay, and mainly the inner membrane have finger-like projections, are called cristae. You can see that. I pointed it there. Now, what are these folds? What do? They increase these folds, because the cristae increase the surface area for ATP molecules generating chemical reactions. Now, ATP, adenosine triphosphate. These molecules play a great role in the chemical activities. They have a great role. Okay, they give energy for all the activities happening in the cell. ATP molecules or ATP, adenosine triphosphate, because they give a lot of energy for the biochemical activities happening in the cell or the chemical reaction happening in the cell. They are known as energy currency of the cell. Okay, the folds, the crystal folds increase the surface area for ATP generating chemical reactions. Means they have a great role in providing energy for all the activities in the cell. So they are known as energy currency of the cell. And it is used for what? For various chemical activities. Whatever the energy they are giving, that is used for various activities in the cell. And that is why mitochondria is known as the powerhouse of the cell. So here ATP plays a great role. It's known as the energy currency of the cell and is required for various chemical activities, and that is why mitochondria is known as the powerhouse of the cell. Here mitochondria stores extra energy, and for whenever the body requires, for the requirement, it stores this energy, and whenever it is required, it is released and is given to the body's requirement. So mitochondria is known as the powerhouse of the cell. This is important. ATP, adenosine triphosphate, it's known as energy currency of the cell and is required for various chemical activities. That is why mitochondria is known as the powerhouse of the cell. So you now understood? Mitochondria, we say it's a powerhouse for the reason for it, because of the ATP molecules or ATP, adenosine triphosphate. They are known as the energy currency. They provide energy for all the activities happening in the cell, for all the various chemical activities. And the structure, as I said, they have an inner and outer membrane. Okay? And they're double-layered membrane, or the inner and outer mitochondrial membrane. And there, the inner folds, they have finger-like projections, right? And they're called a cristae. And that folds of the cristae increases the surface area for ATP generating chemical reactions. Are you all with me? Or relaxing after the small little break you got? Are you with our dear mitochondria? Yes. Yes, somebody said yes. Okay, thank you. Now, mitochondria has its own DNA, deoxyribonucleic acid, and ribosomes. We mentioned ribosomes where it was. Where? In the RER, we say RER is rough endoplasm because of the presence of particles called ribosomes. So mitochondria also has its own DNA and ribosomes, and therefore they are able to make some of its own protein. Mitochondria has its own DNA, deoxyribonucleic acid. You should always know the full form of it, like the ATP adenosine triphosphate. So here, DNA, deoxyribonucleic acid. So they have its own DNA and ribosomes, and therefore they are able to make some of its own protein. When I stress like this, these are features or characteristics of mitochondria important. Normally they ask, draw mitochondria and explain it. 3 marks. Okay. Normally for 3 marks you have to draw and you have to explain the main characteristics. All this will be given in the notes. You don't have to worry. These are the main important ones where I'm stressing. You can hear things what I've said a little here and there. That will be given in the notes. It will be given in such a way that it will be easier for you to study. So I hope you understood about mitochondria. Now we have, I'm sure already you must have read, plastids. We very well know that, we have been studying since last year, okay, that plastids are only present in plant cells. So plastids are only present in plant cells. It's absent in animal cells. When we write the difference, first thing we will say plastids present in plant cells but absent in animal cells. Now here plastids, they are of 2 types. One, chromoplast, colored plastids. Okay, they are colored plastids. They're normally seen where? In leaves, flowers and fruits. Chromoplast, they're normally seen in leaves, flowers and fruits. Like they have a pigment called xanthophyll or keratin, which gives yellow and orange color. You must be wondering, my God, teacher is saying so many things. Don't worry, this all will be given. So chromoplasts are normally seen in the leaves, flowers and fruits. Okay, and because they have pigments called xanthophyll or carotene, which gives color like yellow and orange. As I said, chromoplasts are colored plastids. Now, the next is leucoplasts, white or colorless plastids. They are white or colorless plastids. So here there are two types of plastids. One, the chromoplasts, the colored plastids, and leucoplasts, the white or colorless plastids. I've given a picture here of the plastid. You can see they are also outer and inner membrane. They have also a double-layered membrane, inner and outer membranes. And you can see, I'll be going forward, you can see there's something called stroma. Okay, and you can see, and these structures are called thylakoid. So these are some of the structures. When you're drawing, these are important structures where you have to mark. This is structure for chloroplasts. You should be very careful. You have to draw neatly and label it. That is where marks come on. You can't just draw and just leave it. So here, what I'm saying, you will be having this diagram to draw. That is why I'm stressing on this. When you draw this, you have an outer membrane, you have an inner membrane. Okay, the chloroplast structure it is outer membrane, inner membrane. They are also chloroplast. They have a membrane, double-layered membrane. Okay, and you can see structures. I'll be taking now what is stroma and what is thylakoid. These are the markings. I'll be giving you in the notes a little more easier than this. And you can see these structures are piled up. Coin type of structures are piled up and they're called granum. Okay, they are piled up. Can you see? Like coins, and they're called granum, or they're called thylakoids. This is there. Here now, this is an easy portion. You don't have to worry. It will easily come in your mind. Chromoplast containing the pigment chlorophyll. Obviously, we know. This we have in last we learned. Chromoplast containing the pigment chlorophyll, or the plastids which contain the pigment. That when we learn it, we learn like this: plastids which contain the pigment chlorophyll. That is what I'm sure about. So chromoplast, here in the book it's given, also text is given. Chromoplast, I said they're colored plastids. So here, when they contain the pigment chlorophyll, they are called as chloroplast. Obviously, chlorophyll, it is known as chloroplast. Containing the chlorophyll, it is known as chloroplast. And chloroplasts, obviously, they are important for the process of photosynthesis. Photosynthesis, you have been learning from the 3rd grade, 4th grade. What is photosynthesis? Without photosynthesis, nothing can happen to the plants, so plants can't prepare their food. The process in which they take in carbon dioxide, and by now you should be ready to tell me this definition, even in your sleep also if I ask you, you should be ready. The process by which green plants, they take in carbon dioxide and water in the presence of, what is it? Chlorophyll and sunlight. And whatever they prepare their food is stored in the form of starch, and they release oxygen. Okay? So chloroplasts are important for photosynthesis in plants. Now, chloroplasts also contain yellow or orange pigment. That's what chromoplasts, I said, they are colored pigment. They are colored plastids, and they are found in leaves and fruits and flowers. So they have pigments called xanthophyll and carotene, which gives yellow and orange color. Okay, such things will all be given. Now, here when I said chloroplast is also double-layered membrane. They have an inner and outer membrane. Okay, chloroplastic membrane they have. And here they have inside the chloroplast. Now I'm telling you about chloroplast structure. Just 2-3 lines are there. Here in the text I've just given this much, but I will give you a little more while you write in the note. That is the way to learn. You can't just learn one line. So it's a double-layered membrane. So chloroplast membrane is there. And we have seen inner and outer membrane. And numerous membrane layers are there, and inside a material is present called the stroma. Okay, a material called the stroma is there, and in the stroma there are membrane-bound structures. Okay, the stroma, there are membrane-bound structures which are pile-like coins. Called. You can see they are piled one below the other like coins. And that is known as granum. Okay, they are piled, granum. And each grana Okay, each grana is known as thylakoid. Each unit of grana, single grana, is known as thylakoid. Inside this thylakoid is the pigment chlorophyll that helps in the process of photosynthesis. Okay, the structure will be given. It's not explained here as such. I will give you the notes. So I said chloroplast, they are double-layered membrane. You can see the inner and outer membranes. You can see the double-layered chloroplast membrane. And here we can see they have they are a material inside it called stroma. And inside the stroma, there are, you can see that stroma, the fluid, the material inside the chloroplast. I hope you can see. And inside this, there are membrane structures like this, which are piled, like a pile of coins. You can see, pile of coins called granum. Okay, that is grana. Each unit of grana, a single. It's called thylakoid. And in this thylakoid is the pigment chlorophyll present, which helps in the process of photosynthesis. This is not given in your text as such, but you have to know. So I will give you like how the points which I've said, these points, and for you, like it will be easier for you to learn. Diagram you have to learn, but it won't be as complicated as this. I'll be giving you a simple one where we can mark the stroma, where we can mark the membranes, where we can mark the grana or the granum, and the thylakoid. You can see that. I hope you've understood here this much. So here, I'm just coming up. Chloroplast consists of numerous membrane layers embedded in a material called stroma. We have already said that. And again, these are similar to mitochondria in external structure. So they all are buddies. Okay? They all are buddies. They all are, they all are similar to each other. Somebody is similar to one organ, cell organ will be similar to the other one. Somewhere on the other. Here, these guys, they are similar to mitochondria in external structure. They almost look alike, Maybe their function all is different, but here and there they are similar. So I hope you understood this. I said about chromoplast, okay, and the color pigments, the leucoplast, the white or colorless plastids. And here I said, if the chromoplast, they contain the pigment chlorophyll. We have learned plastids which contain the pigment chlorophyll. The chlorophyll is the same thing. And here chloroplasts are important for photosynthesis plants. I explained, I just gave you a brief, that's more than enough whatever I said about structure of chloroplast. Whichever points are given, that itself will be given in notes also. You don't have to worry, that will be given. But please read the text. And then I've said that about that these are similar to mitochondria in external structure. Here, leucoplast, they have the white or colorless plastid. They play a great important role in storage. They play a great role in storage, mainly in parts like the roots. Okay, their function is mainly connected with storage. Now, leucoplasts are primarily organs, that means very important organs, in which materials such as starch, oil, and protein granules are stored. That is what I said. They play a great role in storage. Are you all with me? Are you? Yes, I heard somebody saying yes. Okay, thank you. And again here, like mitochondria, what is mentioned? I said mitochondria have DNA and ribosomes. So like mitochondria, plastids also have their own DNA and ribosomes. Questions come in this. Name two organelles or name two cell organelles that have their own DNA ribosomes. So you must be ready to write mitochondria and plastids. Okay, leucoplast function storage. They store materials such as starch, oils, and protein granules. It is stored. And here the main, but normally it is the teacher's favorite, as you say. Okay, mitochondria and plastids, they have their own DNA and ribosomes. Now, we're done with our mitochondria. Your favorite, my favorite mitochondria. Then we have done with our plastids. Now, vacuoles. These are nothing new to you. We have learned in short in the 8th. A little more to know this grade. Vacuoles are storage stacks for solid or liquid contents. Vacuoles in plants, they have we know that when we draw the plant cell and animal cell, when we drew the vacuole, there itself we know that vacuole in plants are bigger in size, and animals cell have a smaller in size. When you compare it, we have already learned that when we draw, and then you must wonder, they have a bigger in size, and vacuoles in animals, they are small size. You have to know this. This is the main difference, but you have to make sure they can, true and false. Plants have small vacuoles and animals. You have to be very careful. If you don't know the concept correctly, you might go wrong. Now, in plants, vacuoles are full of cell sap, that is a fluid in it, and provide rigidity and turgidity to the cell. So we have learned that cell wall is the protection layer for the plant cell. So when, in case if the plant cell absorbs water, gets into a process of osmosis, what happens? It will swell and it might turn to burst. But what does it do? The cell wall protects. Okay, it protects from this plant cell to burst. It protects. So that is the portion they are trying to stress, that it provides turgidity and rigidity to the cell. Okay, there's a swelling coming up. Turgidity, mainly it is the part of swelling, that is the part of the swelling of the cell, swells. And rigidity is that it will not allow the cell wall to bend, unbendable. Okay, so here in plants, vacuoles are full of cell sap, the fluid, and it provides turgidity and rigidity to the cell, that is connected with the cell wall. These two words were there last year also, when the cell wall is used for protection and it helps in turgidity and rigidity to the cell. Both, to say turgidity and rigidity, I think you have to twist your tongue and say. Just joking. So here we know, you can see I just given a diagram. Again, obviously when it's bigger, you can see the size is large, so it's understood as a plant cell. And here, these are the main points stressed where you have to learn. And when read the text, again I'm saying read the text, just don't depend on the note. You can underline this. Vacuoles are made of storage sacs for solid liquid contents, difference between them, the plant and animal cell. And in plants, how does it, what is the main criteria for it? That is, vacuoles are full of cell sap. You can see cell sap inside. Okay, it's full of the fluid, and it provides turgidity and rigidity to cell. That is when the if the water enters the plant cell, okay, through the process of osmosis, obviously the cell starts swelling up, and if so, the cell wall protects for protection, so that it does not burst. So the cell, that is why the cell wall is there. We have mentioned this last time, and it provides turgidity and rigidity, the swelling and the unbendable. The cell wall won't bend. Now, in vacuoles, the substances stored in vacuoles include, what are the substances stored in vacuoles? Amino acids, sugars, various organic acids and proteins. So what are the main substances stored in vacuoles? Amino acids, sugars, various organic acid proteins. When I give in points like this, you must understand there will be maybe one line, maybe a true and false. It's obviously there. We can ask, because as I said, we won't be, it's physics, physics, chemistry, bio. So all together comes as one. So we are being divided. That you have been knowing from years now. So it's easier to ask one mark, 2 marks, and then later we have the long answer question, which does not go more, which does not have so much of marks in that. Maybe 1 or 2 questions like that. Otherwise, 1 mark and 2 marks are stressed on. Now, in unicellular organisms like amoeba, the vacuole, the food vacuole, they have a food vacuole. It contains the food items that amoeba has consumed. You must understand that. In single-celled organism like amoeba, the food vacuole contains the food items that amoeba has consumed. So over there, amoeba, happy, the food items are there in the food vacuole. Food vacuole contains food. It makes when it grabs its food, it makes a vacuole and it has the food items in it, and which it consumes it. How does it with this projections called? What is it? Pseudopodia. So it attacks its prey or gets its food. Now another point also. In few unicellular organisms, here amoeba has it like this. Specialized vacuoles removes what? Excess water and waste. Here, like in amoeba, the food vacuole contains the food, whatever it has eaten, has been consumed, it is there, the food items are there in the food vacuole. But in some of the other organisms, there are specialized vacuoles that remove excess water and waste. Just note it down or underline it. Now, the cell organelles are all done. So we've done with all the cell organelles. From the cytoplasm nucleus, we have started from there. Okay, all these cell organelles till now we have stopped till here, the last part of the cell, mainly all the portions till vacuoles. Please go through it. Don't keep it for the last minute to study. I'm not saying that it's immediately study and you do it, but please go through it, learn, plan it and learn. Otherwise all these points won't sit. But I feel the way I've gone with you guys, it should be in your mind. Even if sleep, if I ask you, you should be able to answer. Somebody's smiling. Teacher will ask me to sleep now. Here, each cell. Now the importance of this. Each cell acquires a structure and ability to function because of what? Because of the organization of membrane and the organelles. So each cell, whatever structure acquires, the structure and the ability to function. Why is able to function properly, or the structure what it has? Because of the organization of its membrane and the organelles. Now, organelles help the cells to perform functions like we have learned this, different organelles, different functions. Just giving a recap: respiration, obtaining nutrition, clearing of waste materials, and forming new proteins. Thus, cell is the basic functional unit of life. Hope you have understood this. This is a general thing, but you should know what in general is 1 mark. What does the organelles help to perform function? What is its importance? What is important of the organelles? So organelles of the cells help to perform functions like respiration, removal of waste or clearing of waste, obtaining nutrition and forming new proteins. So they can ask this one line. So thus cell, and I've given a question, why is it called basic and structural unit of life? I think by now you must be knowing this. Here, cells will grow old, will die, it will get injured, and it will be replaced by, by new cells, by marking growth, by growing. And another thing, and the gametes required for reproduction does happen, are also formed from new cells. We know that cells will die, they'll grow old, they'll get injured, they are replaced by new cells, okay? And marking growth, they grow. And the gametes, even for reproduction, gametes are formed, and they are all formed from the new cells. So such process where new cells are made, such things when it's happening, or such process when it's happening, the process by which new cells are made is called cell division. So what is cell division? The process by which new cells are made is called cell division. Important. You have to know or define what is cell division. The process by which new cells are made is called cell division. Now, cell division are of 2 types. Cell division you might be knowing. It's not new. Maybe a few of you might be knowing cell division. But now cell division of 2 types, because I have mentioned 2 things up here. The cells, cells grow or die and injured and will be replaced by marking new cell growth. Here one growth, and here the gametes required. So 2 points have been mentioned. There's a reason for it. Now cell division is of 2 types, that is mitosis and meiosis. Okay? There are 2 types of cell division. Here we're just going to have a brief about it. Higher class when you go, you can elaborate. There's so much to know about this, the mitosis and meiosis. Okay? So meiosis, I hope it's okay. Okay. Mitosis and meiosis. When you go in the higher classes, it's given in elaborate, in detail. Here we just have to know a little of it, what it is, and the function mainly. That's all given in your text. So there are 2 types of cell division, that is mitosis and meiosis. Now what is mitosis? The process of cell division by which most of the cells divide for growth. The cell which divides mainly for growth. The process definition, you have to write it correctly, my dear children. It's very important. So you can say cell division, it's divided into 2. Okay, a cell divided into 2. So the process of cell division, the cell division happening here. Are you all with me? Okay. By which most of this cell divides for growth is called mitosis. You can see a single cell divided into two. Now in this process, each cell is called the mother cell and to form the two identical daughter cells. Here we don't say father cell and son cells. We don't say all that. Here we say it's a mother cell, divides to form two identical daughter cells. Okay? I am sure we have gone through this earlier, a bit of it, but we have not said like mitosis or meiosis. So you must understand, in this process, cell division, okay, a mother cell is divided into two identical daughter cells. For heaven's sake, don't write father cell and son cell and give me. Be very careful. Okay, in this process, each cell is called a mother cell, divides to form two identical daughter cells. Now what is it? The daughter cells, those two daughter cells in the picture, have the same number of chromosomes as the mother cell. Important. Okay? They have the same number of chromosomes. Chromosomes found in the nucleus, right? When the cell is not dividing, the chromatin material and it condenses to form when it's not dividing. It's chromatin material. When it starts dividing, chromatin material becomes chromosomes. And the main interaction point of feature there, who? The genes play a great role there. That is for inheritance, transfer of characters from the parents to offspring. This all you know. I'm just brushing up or giving you a record. Now, what is the use of mitosis? As I said, a brief. What is mitosis? You must know a little about its process. And what does it help into? Mitosis helps in growth and repair of tissues in the organism. What is the function? Mitosis help in growth and repair. Obviously, like if your car is damaged, what do? You repair it. You go where? To the workshop and get it repaired. The same way, these guys play a great role. Mitosis process helps in the growth. Normally, they have to go in the growth and the repair of tissues in the organism. I think this is not that difficult. The mother cell divides into two daughter cells, two identical daughter cells, and whatever daughter cells will get the same number of chromosomes as the mother cell. That's important, you have to know. And what is the function of mitosis? It helps in the growth and repair of tissues in the organism. Now here we're going to the next process. Now, specific cells of reproductive organs or tissues in animals and plants divide to form gametes. You know what is the gamete? Gamete for us, the male gamete, the sperm. The female gamete is the ova. Okay. Here, what happens? Fertilization. What is fertilization? Yeah, the fusion of, somebody is saying it. Yeah, fusion of the male gamete and the female gamete to form a zygote. Okay. So here, the process of meiosis is taking place here. So specific cells of reproductive organs or tissues in animals and plants, they divide to form gametes, which after fertilization, obviously after fertilization, they give rise to offspring. You can see a cell dividing two. Till here we know. That is the mitosis. But now these two are divided into further division is happening. You can see 1, 2, 3, 4 of them. The division is called meiosis, which involves two consecutive divisions. That means once the cell it divides in two, again those two will be further divided. When a cell divides by meiosis, it produces 4 new cells instead of just 2. Again, I am stressing. Mitosis have 2. Here in meiosis, it is 4 new cells instead of just 2. Please don't mix up and write. Mitosis, they have 2. I said a mother cell divides into 2 identical daughter cells, and the chromosome number, whatever the mother cell is having, the daughter cells all will have the same number. Okay, that is why I'm stressing. This is important. Differences when it might come mitosis and meiosis. You have to know. You have to obviously when you write differences, define mitosis and define meiosis. And these are the points where you have to stress. Here mitosis it's 2, but in meiosis they produce 4 instead of just 2. And what happens? The new cells will only have half the number of chromosomes than that of the mother cells. The other one, they have the whatever chromosomes in mitosis the mother cell has, they will get exact number. Again, I am saying, in mitosis, whatever chromosomes, the number the mother cell is having, that chromosomes exactly will go to the two daughter cells. That is, the identical daughter, they will receive the same number as the mother cell. Here, they will get the new cells which have been formed or will have only half the number of chromosomes than that of the mother cells. That is the main difference. And here I said meiosis, it is 4. They produce 4 new cells instead of just 2. Hope you have understood. Cell divides into 2. Here, till here it is mitosis. That's what I have explained. A mother cell divides into 2 identical daughter cells, and whatever chromosome number the mother cell gets, it will be, they will get it. The daughter cells will get. But here in meiosis, they will get the 4. 1, 2, 3, 4. They will get only half the number of chromosomes than that of the mother cells. This is important. Normally questions come here, the function, what is it, mainly I said the mitosis, what is function, mainly stressed here is, write the differences between mitosis and meiosis. 2 marks, okay, you can just define and give, and then, and you can just, even you can draw the diagram also, inside the differences, it will be more easy for us also to look. Then, so with that, I am done with this chapter. We had a long way. Literally, we were inside the cell with our dear cell organelles. We were there. We learned so many things. We learned about our dear scientists. From there, we started who all discovered what. We learned all that. Those scientists never, please don't forget that portion is important. Then we went around the characteristics of the cell. Okay, those also important. We went around all that, and then we started with the osmosis, diffusion, where that is mainly in the plasma membrane. Remember why I'm telling this? So that now it will, you can recall, or somebody's, now I think somebody's turning the pages. Yes. And then we went around there. Then I've told you the different types of solutions, the osmotic, how it is, the osmosis and the diffusion process. Then we went around saying, learning about the cell wall, then we went nucleus, then we went for our ear, then a bit of cell organelles were there. You must be wondering, my God, teachers by heart in this. Yes. Ear, then the Golgi apparatus, lysosomes, mitochondria, plastids, vacuoles. These, everything is important. I've tried my best to make it easier for you to study.